Hello everyone, welcome to ITLS Academy. Our today's lecture is about the human embryonic stem cell and the human embryonic germ cell. We will discuss all these two topics in detail in this particular lecture. So guys, let's start. First of all, we will see an overview of embryonic stem cell and embryonic germ cell. Very first point is in 1998, James Thomson and his colleagues reported methods for deriving and maintaining human embryonic stem cells from the inner cell mass of human blastocyst. Blastocyst, we have discussed what is blastocyst in detail in our previous lectures. If you have not watched, then please go through those lectures and see what is blastocyst. For reference, I can tell you that blastocyst is something like this structure. This is formed after the formation of zygote. This is a developed stage. It contains a trophoblast layer, this is trophoblast, and also the inner cell mass. This is the structure of blastocyst. So these scientist studied, worked on this blastocyst, and that were produced through in vitro fertilization and donated for research purpose. Embryonic stem cells are stem cells derived from the undifferentiated inner cell mass. You can see this is inner cell mass, and embryonic stem cells are derived from this particular inner cell mass. Embryonic stem cells are derived from the inner cell mass. Embryonic stem cells are pluripotent, meaning they can give rise to a multiple type of cells. Next point, these cells were cultured from primordial germ cells obtained from the gonadal ridge and mesenchyma of 5 to 9 week fetal tissues that resulted from electric abortions. The two research teams developed their methods for culturing human embryonic stem and embryonic germ cells by drawing on a host of animal studies. Here you can see a detailed structure of a human embryonic stem cell. This is embryonic stem cell. Let's see the formation of this particular cell. This is blastocyst embryo. This structure is a blastocyst embryo. This is formed after the formation of zygote. If we discuss, this is egg and this is sperm. After their fusion, they forms zygote. This structure that is known as zygote is a type of cell that can give rise to whole body. Whole body can be derived from this zygote. So it is also known as totipotent cell. Totipotent means a cell that can give rise to all the kind of cells. So the zygote is a type of cell that can give rise to all of the kind of cells. When this zygote differentiates, it divides from 1 to 2, 2 to 4 and so on. After many divisions, a stage forms known as blastocyst. This blastocyst is a stage at which implantation of this zygote occurs. Just before the implantation, that is attachment to the ovary wall, is known as blastocyst. This is the stage from the point the zygote attached to a uterine membrane and further starts development. So, when this blastocyst develops, after the attachment, development of this blastocyst starts and this forms blastocyst embryo. This blastocyst embryo have two parts. This is, you can see, this is outer layer. This outer layer is known as trophoblast, while this is also have a mass of cells. There are various cells gathered at a place. This is known as inner cell mass, or in short we call it as ICM. This inner cell mass, as this whole process we are studying in vitro. So in vitro culture is a culture in which the conditions for growth and development are provided artificially. If we talk about the natural environment, this blastocyst gets its nourishment from the mother womb. But when it comes to in vitro, it requires culture of cells. So, culture of this blastocyst embryo cells is done. After the culture, it forms embryonic stem cell. As we know that these embryonic stem cells are pluripotent in nature. These are pluripotent, so these cells can give rise to many kind of cells. Here you can see this is unlimited growth. This can give rise to unlimited type of cells. Further it goes to differentiation and forms. You can see these are neuron cells, these are RBCs, and these are muscle cells. And many more kind of cells are produced by the embryonic stem cells. These embryonic stem cells cannot give rise to trophoblast. Except trophoblast, all these cells can be formed from the embryonic stem cells. So that's why embryonic stem cell is known as pluripotent cell. Here is timeline of human embryonic stem cell research. We will see what is studied in previous years to get final outcome of these embryonic stem cells. A long research has been done on the human embryonic stem cells. So we will see year wise what research has been done in a particular year. So in the 1878, first reported attempts to fertilize mammalian eggs outside the body. First attempt to fertilize egg outside the body is done in 1878. In 1959, first report of animal, and uh, if we talk about specialty, then this animal was a rabbit. So first report of animals produced through IVF, in vitro fertilization, that is outside the body, in the United States. So first production of animals through in vitro fertilization, or we can say that first animal production outside the body has been done in 1959. In 1960s, studies of teratocarcinomas in the testes of several inbred strains of mice indicates they originated from embryonic germ cells. There was a study on teratocarcinomas, and that was totally based on indicating that these several strains of mice originated from the embryonic germ cells. In 1968, Edward and Bavister, two scientists, fertilized the first human egg in vitro, that is, first in vitro, first from outside the body fertilization of a human egg was done by Edward and Bavister in 1968. 1970s. Embryonic cells injected into mouse blastocyst produce chimeric mouse. Chimeric is a term that means there is combination of any two things. In 1978, Louis Brown, the first in vitro fertilized baby, is born in England. These are some important terminologies that you can remember. That first in vitro fertilized baby named as Louis Brown, and he was born in England. In 1980, Australia's first in vitro fertilized baby, that is Candace Reid, is born in Melbourne. This was second baby that is formed by in vitro fertilization. In 2000, scientists in Singapore and Australia, led by Pera Tronson and Bongso, derive human embryonic stem cells from the inner cell mass of blastocyst donated by couples undergoing treatment for infertility. When it comes to 2001, As human embryonic stem cell lines are shared and new lines are derived, more research groups report methods to direct the differentiation of the cells in vitro. Let us see the derivation of human embryonic stem cell. The first documentation of the isolation of embryonic stem cells from human blastocyst was in 1994. Since then, techniques for deriving and culturing human embryonic stem cells have been refined. Embryonic stem cells come from human embryos that are 3 to 5 days old. They are harvested during a process called in vitro fertilization. This involves fertilizing an embryo in a laboratory instead of inside the female body. The embryonic stem cells used in research today come from unused embryos. These result from an in vitro fertilization procedure. They are donated to science. Here in this diagram, you can see human blastocyst showing inner cell mass and trophoectoderm. This is trophoectoderm layer, or it is also known as trophoblast layer, while this is a mass of cells known as inner cell mass. This is picture of particular human blastocyst. This particular picture was in vivo blastocyst. Now we are going to see blastocyst in vitro, that is outside the body. In this diagram, you can see male and female pronuclei approach each other, that is fertilization process. Egg and sperm comes together and fertilize. Next comes first mitotic division, and these divisions continue till the formation of blastocyst. There are multiple divisions, and these divisions in the case of zygote are known as cleavage. You can see this is forming 2 cells from 1 cells, 4 cells from the 2, and this goes on continuously, and finally there is formation of blastocyst. Here an intermediate stage of the mass of cell comes, that is known as morula. In humans, blastocyst formation begins about 5 days after fertilization, when a fluid-filled cavity opens up in the morula. You can see this is cavity, and this cavity is known as blastocyst. When this is morula stage, when there is a formation of cavity in this morula, then this is known as blastocyst. So it comes after when a fluid-filled cavity opens up in the morula, the early embryonic stage of a ball of 16 cells. The inner cell mass begins to separate from the outer cells, which become the trophoectoderm that surrounds the blastocyst. This represents the first observable sign of a cell differentiation in the embryo. Many in vitro fertilization clinics now transfer 5-day embryo to the uterus for optimal implantation. Implantation is a stage of development that more closely parallels the stage at which a blastocyst would implant in the wall of the uterus in vivo, that is inside the body. Derivation of human embryonic germ cells. Human embryonic germ cells are derived from the primordial germ cells, which occur in a specific part of the embryo or fetus called the gonadal ridge, and which normally develop into mature gametes, that is, egg and sperms. Embryonic germ cells are pluripotent stem cells derived from primordial germ cells. What is pluripotent we have discussed in detail in our previous lecture. Embryonic germ cells are the cells in the embryo that give rise to the reproductive cells, that is, gametes, of sexually reproducing organisms. In animals, male gametes are sperm cells and female gametes are egg cells, also known as ova. In this particular slide, very basic things are discussed so that you can understand how the things are going. Here you can see this is process that is occurring in testes, and this is process that is occurring in ovary is shown here. In the testes, first cell, that is gonocyte. This gonocyte forms spermatogonium. This spermatogonium produces primary spermatocyte, and this primary spermatocyte forms secondary spermatocyte. This secondary spermatocyte forms spermatid, and this spermatid forms mature sperm. Let us stop at this point and let us see what is happening in the ovary. In the ovary, this is gonocyte. This gonocyte forms oogonium. This oogonium forms primary oocyte. This primary oocyte goes further development and forms secondary oocyte. Secondary oocyte forms antral follicles, and these antral follicles finally forms mature ovum. Now you can see here this is mature sperm and this is mature ovum. Here comes the process of fertilization, or that is meeting of these mature gametes. When both of these gametes fuse together, these forms one cell embryo. These two cells form one cell embryo. This one cell embryo further goes to development and forms blastocyst. This blastocyst formed after the many cleavages. After many cleavages, this blastocyst formed, and this contained a trophoblast outer covering and inner cell mass and a cavity that is known as blastocoele. This blastocyst further goes in the process of gastrulation. After the process of gastrulation, you can see there is formation of PGCs, that is post-gastrulation cells. These are post-gastrulation cells and further goes to the formation of further cells. You can see that these cells can further form testes and ovary, and this process is repeated. If we talk about the culture of blastocyst in vivo and in vitro, you can see in vivo, that is inside the body, this is blastocyst. This blastocyst forms primitive ectoderm or epiblast, and further it forms embryonic stem cells. These embryonic stem cells forms post-gastrulation cells. These post-gastrulation cells forms somatic stem cells and oocyte, that is germ cells. And after the fertilization, again this process is repeated. This process is inside the body. This is happening inside the human body. So this is known as in vivo. If we talk about the in vitro, how this process is carried outside the body, you can see here these are somatic cells. These somatic cells form iPSCs. This is the culture. After the culture, these forms embryonic germ cells. Embryonic germ cells are formed. Also here the formation of post-gastrulation cells or post-gastrulation like cells. Like, that is, here it is shown that star mark points at can form teratoma in vivo. This is blastocyst. This forms primitive ectoderm or epiblast. Further it forms embryonic stem cells, and these embryonic stem cells also forms post-gastrulation cells. When these both comes together, these forms somatic stem cells, sperms or oocytes. This blastocyst fuses to embryonic germ cells. This is the whole process how all these things happens outside the body, that is in vitro. There was a scientist, that is Gerhardt, and his collaborators devised methods for growing pluripotent cells derived from human embryonic germ cells. You can see here human embryonic germ cells. The process requires the generation of embryoid bodies from embryonic germ cells, which consist of an unpredictable mix of partially differentiated cell types. If we talk about post-gastrulation cells, so these cells are progenitors of adult gametes, which diverge from the somatic lineage between late embryonic to early fetal development. These post-gastrulation cells first derived in the mouse. If we talk about embryonic germ cells, these cells have also been derived from humans, chicken and pig. So thank you guys for watching this session. Follow ITLS Academy on various social media platforms and also subscribe ITLS Academy, that is our channel on YouTube. Thank you very much for watching.